So now we're going to get into the urinary system. With this particular system, we're really going to put a major focus on one of the organs in the urinary system, and that is the kidney. But we are going to go over the major roles that the renal system or the urinary system plays in general. We'll talk about some of the anatomy that is relevant. We'll talk about how the kidneys are supplied in terms of the nervous system as well as vascularly. We'll get into the nephron. We'll get into the renal corpuscle and filtration member, the juxtaglomerular apparatus, and the renal tubule and all the different types of cells we find along that, and then getting into basically the difference between cortical and juxtamedullary nephrons, and then talk about the peritubular capillary and the vasa recta when it comes to the nephron. So if we look at the overview of the renal system or the urinary system, we're really putting a major focus on the kidneys here because they're the workhorse when it comes to maintaining homeostasis. It's really one of the major things that they do is really to promote homeostasis systemically. And they're going to achieve that by helping filtering the blood as well as helping with the formation of urine, which is going to be a nitrogenous waste product, with other things as well. But this is really simplified because in order to filter blood and make urine, we are going to be regulating the pH of the blood, right? How much bicarbonate, how much hydrogen ions do we allow in there? Regulating blood ion concentration, so electrolytes, sodium, potassium, chloride, blood osmolarity, and blood volume and blood pressure. Also with certain hormones like calcitriol, EPO, renin, these are going to be coming from the kidneys. And then also some gluconeogenesis if we are under starvation conditions. And then when it comes to waste, right, getting rid of mainly nitrogenous waste as well as any sort of metabolites that we need to get rid of, such as the breakdown of drugs or toxins. The others that help with this include the liver, the lungs, the sweat glands, and the GI tract. They will assist with this. So for example, the liver can help with the breakdown of waste and drug products and whatnot, and then to a point where the kidneys can easily filter it out. So urine is made inside the kidney, inside of these microscopic structures in the kidneys called the nephrons. These nephrons, right, again, are these little microscopic guys, but they're going to be found, generally speaking, kind of along the renal cortex and medulla, kind of riding that line. The urine is going to be made with those nephrons. It's going to then drain into all of these little renal pyramids, right, which then continue down, right, from that renal papilla of a pyramid downward into these collecting ducts, which then all join together to form the renal pelvis, which is this structure here, right, coming out of the hilum. And then that is going to be continuous, right, that renal pelvis is continuous with the ureter. And so we take that urine down through the ureter to the bladder. The bladder can store that until it's appropriate to eliminate that urine, and then it can transport the urine from the bladder to outside of the body via the urethra. And we're using peristalsis, so there's some smooth muscle involved here to help contract it down. We're not just relying on something like gravity. So there's a couple main parts, some of them already named. When we look at the kidney, namely the renal medulla and cortex, the medulla is this inner region, think middle. Renal cortex is the outer layer. The renal medulla is made up of these pyramids, which are where we can do things like secrete and absorb, and the calyces, which are going to help funnel that urine towards the pelvis, right? We see them, these kind of larger, almost tubular shaped structures, and then the ureter, again, just from the renal pelvis, is continuous down to the bladder. But let's talk about the nephron, because the nephron really is the one we care about. We have over 1 million of them in each kidney, right? Roughly. These are these microscopic structures that are going to help with this filtering process. And we have a few main components. We have the renal corpuscle, which has two components to it. We have the glomerulus, which is this kind of capillary bed or capillary knot found inside of there. And then we also have a capsule surrounding it, sometimes called a glomerular capsule, sometimes called a Bowman capsule because Bowman found it. But the two of those things together we call the renal corpuscle. From there, we have the proximal convoluted tubule, or the PCT. It's called that because it is going to be, in terms of the flow of fluid, it's closest to the renal corpuscle, and it's convoluted, so it's all twisty and turny, right? Then we have the loop of Henle. Sometimes this is also called the loop of nephron, or the nephron loop. I will use loop of Henle because that's how I learned it, right? But if you use those, I'm okay, right? If you use those particular, any of those other terms. So the loop of Henle is going to dive downward, right? Loop back up, around, and back upward. And then we have the distal convoluted tubule here, which is going to, again, be topsy-turvy, twists and turns, right? All sorts of stuff. Now notice here that it does kind of touch, right? This distal convoluted tubule does kind of touch right between where the afferent and efferent arterioles are. So it's a little bit, and this is going to be important. We'll get to it in a little bit. We're not going to worry about it right now, but that's important. Keep a note, even though it's touching, it's still distal, because the fluid that's found in the distal convoluted tubule had to go through all these other parts, right? It's towards the end of the line, which is why we call it distal. And then the distal convoluted tubule will join up with a collecting duct, which is connected to several of these nephrons, which will then take us down towards that papilla of the renal pyramid, down towards those calyces, and towards that renal pelvis. And so if we look at the formation of urine, it really comes down to 3 steps, right? And it's all going to come down to bulk flow, active transport, and diffusion. Some things are going to we have bulk flow, we are using pressures, to like hydrostatic and osmotic pressures to move things, like to move massive amounts of fluid. We have active transport to actively, use energy to bring things back in or push things out. And diffusion, we're using just letting things just, pass through using those concentration gradients without any energy input. But the 3 steps of forming urine is glomerular filtration, tubular reabsorption, and tubular secretion. Now I'm just going to call them filtration, reabsorption, and secretion because, as you can imagine, these steps are a mouthful. But for right now on this slide, I'll use their full names. Glomerular filtration is where we are taking solutes, we're taking solvents out of the capillary. So out of that glomerulus into the capsular space. Tubular reabsorption, we're taking solvents or solutes back into a capillary. And then tubular secretion is similar to filtration. Solvents or solutes are making their way out of a capillary. Now there's 2 different kinds of nephrons, and they make 2 different types of urine: cortical and juxtamedullary nephrons. Cortical nephrons are the majority, and if you notice, for the most part, they're hanging out in the cortex, right? Which is why they're called cortical nephrons. So most of the nephron is hanging out in that cortex, and just a little piece of the loop of Henle goes down into the medulla. And this is where we're making urine with osmolarity that's similar to blood, right? So we're going to have a similar kind of, concentration to what blood would have, which means mostly water. This is going to be most of your nephrons. Then the juxtamedullary nephrons, this is where that long nephron loop dives deep down into the medulla in comparison. So we see, like up here, most of that loop of nephron is still in the cortex with just a little section down in the medulla. Over here we see almost that entire loop, pretty much that entire loop, is going down into that medulla. And so these guys, they're going to be producing highly concentrated urine. But let's go ahead and talk about this renal corpuscle and talk about what's going on here, because this is where we're going to have filtration happen. It's going to be happening in this renal corpuscle. So the two parts, I already named them, but we'll go over them again. We have the glomerulus and the glomerular capsule, also called the Bowman's capsule. I'm just going to call it Bowman's because it's easier to say. But the glomerulus, this is going to be this kind of like almost like a capillary knot. It's what we call a tufted capillary. It is made of fenestrated capillary type, is a fenestrated capillary type. And this is going to be fed, right? We have arterioles that are going to enter in and leave in this glomerulus. The afferent arteriole arrives. It's going to bring blood to the glomerulus. The efferent arteriole exits. That's the way I remember. Afferent arrives, efferent exits. So efferent arteriole will exit out, right? Continuing on. The Bowman's capsule, right, is going to have this layer, right, what we call a visceral and a parietal layer. Visceral being deeper in. This is what's going to have direct contact with the glomerulus. And then the parietal layer, which is going to be forming kind of the outer edge, right, the capsule itself. The visceral layer is going to have what we call podocytes. That's these guys here. They almost look like little serrated cells kind of all over the place. They have all these little extensions. They wrap around those capillaries, and they're going to be part of that filtering system, right? So we're not only going to be filtering out just using, like how capillaries would, we're also going to have these extra elements added in. And this is where we get our early filtrate. The early filtrate gets collected between the visceral and the parietal layers. We're looking at about 180 liters a day. Now, obviously, you don't urinate 180 liters a day. Almost all of this is returned back to the blood. Almost all of this. But a lot of it's going to leave here, right? A lot of that early filtrate is going to have a lot in it. And it's as we travel through the rest of the nephron that we will reabsorb a lot of this to put back into the bloodstream. And if you remember, I talked about that area where the distal convoluted tubule touches right between the afferent and efferent arterioles. And I mentioned it's called the juxtaglomerular apparatus, or the JGA. This is going to be a special little location. We have these specialized cells like the macula densa that are going to be able to detect things like solute concentrations. And this is going to help us when it comes to maintaining things like blood pressure. So in this macula densa, right, if we look here is like a cross section of that distal convoluted tubule, right? So we're looking inside the open space of that tubule, and we see that there are these specialized cells right where we touch between the efferent and afferent arterioles. And these guys have osmoreceptors. So they're going to detect changes in solute concentration, so that as we're getting towards the end, right, of that nephron, the filtrate passing through, we can detect what solute concentration is still left, and then that will cause these juxtaglomerular cells to release a hormone called renin. And renin is going to activate the RAA system. The RAA system being the renin, angiotensin, aldosterone system. And so that's going to be helpful when it comes to maintaining blood pressure. And we'll get into what renin, angiotensin, and aldosterone do in just a little bit. But we find them there. So these cells on the afferent arteriole, right, they have very special cells containing renin. They will sense blood pressure. So we have some that detect the solute concentration inside of the filtrate. And then these JG cells that also detect the blood pressure because they're mechanoreceptors. So they detect the pressure being exerted on them. And they can release that renin when they are stimulated to do so. Now let's go ahead and talk about the rest of this. We got through the renal corpuscle. Now let's talk about the renal tubule. The renal tubule, right, is going to be basically the rest of it. which is going to compose of the PCT, or the proximal convoluted tubule. We're going to have simple cuboidal cells here. They're going to reabsorb most of the filtrate. So a lot of stuff gets filtered out, but then very quickly gets pulled right back in into the bloodstream. The loop of Henle, which is going to be that loop that dives down. We have a thin descending section, which is made of simple cuboidal for absorbing water, and then we have a thick ascending section, which is columnar. That's what's going to make it a little thicker, because we're columnar cells instead of cuboidal, and that's going to help us with reabsorbing things like sodium and chloride. So it's going to help us with absorbing salt, right, basically. So we reabsorb water, and then as we come back up, we reabsorb salt. Now, as we enter into the distal convoluted tubule, we're back to thick, simple cuboidal cells. We're going to reabsorb more sodium. We're also going to reabsorb calcium, and this is in response to our parathyroid hormone. And then when we get towards the end of the DCT and we get towards the collecting duct, we have principal cells that reabsorb sodium. We have aldosterone receptors, which are going to respond to that hormone aldosterone to help us reabsorb sodium as well as water, and to get rid of potassium. And then ADH receptors, which is antidiuretic hormone receptors. These will promote the water reabsorption. We also have intercalated cells, which will help with blood pH. And so type A cells, they will basically get rid of hydrogen ions or protons and keep bicarbonate. So we're going to put the protons into the urine. We're going to keep the bicarbonate in the body with type A. Type B is the exact opposite. It's going to get rid of the bicarbonate and keep the protons. And so if things are too acidic, then type A cells kick on, right? Think type A will handle the acidic environment to bring us back to normal. Type B cells, right, they're going to handle when things are too basic. So type A cells handle things when things are too acidic. Type B cells handle things when things are too basic. And maybe that'll help you remember that, because they're A and B, right? So if we take a look at what is going on throughout this entire tubule, we have the simple cuboidal cells reabsorbing over half of what even filtered out in the first place, because a lot of it is like water and glucose and all that kind of stuff, all sorts of fun stuff that we still want. Then as we travel down to the loop of Henle, as we're going down, we're going to reabsorb water. As we come up, we're going to reabsorb salt. And then as we enter the distal convoluted tubule, in the early part, we're going to reabsorb sodium and calcium, and as we get towards as we get to the late distal convoluted tubule, we will worry about pH with type A cells when things are too acidic, and type B cells when things are too basic. And then as we continue down, getting further and further, we then get we have a lot of hormonal influence, namely aldosterone promoting the reabsorption of sodium and the secretion of potassium, and then ADH promoting the reabsorption of water. Now, in terms of the vascular nerve relations, the kidneys have a very nice, strong blood supply via the renal artery and renal vein. Basically, these renal arteries come right from the descending aorta, which means a really good blood supply, and this is going to allow the kidneys to really be efficient with filtering blood. And then we connect to the inferior vena cava with the renal veins. So the blood that's going to the kidneys really is it's fresh, it's straight from the aorta, and then we're pumping it right back in towards the heart. So it's a really effective way that we're not going to have a ton of stuff in the way of that pathway. So the kidneys are because of this connection too, they're 0.5% of the body mass that you have, and yet they get about a fifth to a quarter of the resting cardiac output. And again, they're doing all this waste removal. They really are the main organ responsible for this waste. Like I said, the liver it will help break things down, but it doesn't get rid of it. The lungs will help with getting rid of things like carbon dioxide and help a little bit with acid-base balance, but not as strongly, not enough, right? So the kidneys really are the ones that are handling things like the pH balance. They're also the ones who are actually doing the eliminating, the actual real filtering of nitrogenous waste products, of which there are a lot. That is something we do make a good amount of. And so it makes sense that they would receive so much blood, that they would have the blood setup that they do. With the nerve supply, the renal nerves basically are carrying autonomic nervous system sympathetic signals. So we have sympathetic nerve fibers that are going to travel along the renal artery. And basically this will help regulate the blood flow through the kidneys, which will then influence urine production. So as sympathetic stimulation goes up, blood flow increases, which means we produce more urine because there's just more blood flowing through, more filtrate being produced. And now the nephron forms urine by exchanging substances between the filtrate and what we call the peritubular capillary. So you've probably been thinking, okay, we're reabsorbing things and whatnot as we go down like this loop of Henle and whatnot, but where the heck does it go? I don't see any blood vessels. And that's because in the previous pictures we had eliminated that. We hadn't really been showing that, but we have this kind of capillary bed that is woven around the proximal and distal convoluted tubules as well as the loop of Henle. Right, and so this is what we can reabsorb into, right, is this peritubular capillary. This is going to be fed by the renal artery and is going to empty into the renal vein. And then the vasa recta is going to be just the section of it that will dip down along that loop of Henle.